Welcome back to Digestive System Part 2. Looking at the small intestine. This is the site of greatest absorption of digestion and absorption. The divisions are going to be the duodenum, which first comes off the stomach, and it's a short piece. Then the jejunum, which is the longest part. And then finally, the ileum. And within the ileum, if we look at it under the microscope, we're going to see what are called Peyer's patches, which are lymph nodules, basically. As you can see, the ileum is going to attach to the colon at what we call the ileocecal junction. You'll see an ileocecal valve within the ileocecal junction. And there is a lot of bacteria, of course, in the large intestine. And so these Peyer's patches help to prevent that bacteria from entering into the bloodstream. And some of the modifications that we're going to see, there's going to be circular folds, which are called the plica circularis. We're also going to see villi, which are finger-like projections. We'll see a lacteal. This is some lymphatic vessel that is going to be found within the villi. And again, this is for absorbing certain fats. And then we're going to see microvilli. So let's talk about that for a second. We have microvilli, if you remember from last semester. These are extensions of cytoplasm off of the cell, typically the columnar cells. And their function is to increase surface area. Okay? So the more surface area you have, the more contact you have with the foodstuffs, and the more nutrients can be absorbed through those microvilli. Okay, so that is going to be for increased surface area. Then we have a villus. A villus is a finger-like projection, and it's going to have the columnar epithelium attached to it all the way around. And its function is to once again increase surface area. Okay? And so we're going to have millions of these villi. Again, these finger-like projections, increasing surface area. And then we have the plica circularis, the folds. What does that do? You guessed it. It increases surface area. So we have surface area upon surface area upon surface area to try to absorb as much as we can nutrition-wise from the gut. Now the cells of the mucosa are going to be absorptive. They're going to be goblet cells, and if you remember, goblet cells make mucus. And there's going to be granular and endocrine cells there as well. Now going back to the parts, duodenum, jejunum, ileum. So DJI. Wait a second. I is what in Roman numerals? Oh yeah, it's number 1. DJ. Oh, I know. Dr. Jerry. So if you want to remember this in order, Dr. Jerry is number 1. Numero uno. Ichiban. Okay, you got the point. Anyway, so that's one way of remembering that in order. DJI. Okay, so the small intestine secretions. Again, there's mucus. And the mucus helps protect against digestive enzymes and stomach acids. Because the things that these things are breaking down, well, the intestines are made out of it. Okay? Such as proteases break down proteins, right? Also, the stomach acid denatures proteins. So you have to have this mucus barrier to prevent from digesting the stomach or the intestines. The digestive enzymes, we have disaccharidases, and anytime you see ACE, remember that it's either an enzyme or a hardware store. In this case, it's an enzyme. Okay, they're spelled differently. But anyway, the disaccharidases help to break down disaccharides into monosaccharides. And those monosaccharides are what we're going to utilize for energy. So, mainly glucose. We have peptidases. These hydrolyze peptide bonds. Well, where do you find peptide bonds? We find those in proteins. So basically, the peptidases are going to break proteins down into eventually their individual amino acids. And then we have nucleases, and those break down nucleic acids. Well, where do we have nucleic acids? Well, we have DNA, which is deoxyribonucleic acid, and we have RNA, which is ribonucleic acid. So we break them down into the individual nucleic acids so that our body can utilize those to make more DNA and to make more RNA. And then duodenal glands, it's stimulated by the vagus nerve. The vagus nerve has a parasympathetic action to it. And so it's also stimulated by secretin, also by chemical or tactile irritation of the duodenal mucosa. So let's take a look at the duodenum and the pancreas. The stomach would be attached here. And so here's the first part of the duodenum. Now we have the pancreas that kind of sits right in the bend or curve of the pancreas. And what you'll notice here is the common bile duct coming down. It's attaching to the pancreatic duct in what we call the hepatopancreatic ampulla. So it's a little swelling there, that hepatopancreatic ampulla, of where these two combine. And then they're going to poke out a bit through the duodenum. And if we run a scope through there, we'll see a little bump there. And that's the major duodenal papilla. Okay, so major duodenal papilla. And then there's a little sphincter right there that controls when bile or the digestive enzymes from the pancreas are going to be released into the duodenum, and that is called the sphincter of Oddi, ODI. So sphincter of Oddi. Okay. And you can see where this could be a problem. If we have a gallstone that you pass, and suppose that gallstone lodges right here in the hepatopancreatic ampulla, that can cause a backflow of the digestive enzymes from the pancreas, and you can wind up with some severe pancreatitis. Okay, because they don't always make it all the way through and pop out through the duodenal papilla. Okay, we can see some circular folds in there as well. And while I'm on the pancreas here, I'll talk about that this is the head of the pancreas. This is the tail of the pancreas. And then in between is going to be the body of the pancreas. And so what else? Now here's an accessory duct that we often see. And again, that will poke out into the duodenum. And we'll call that the minor duodenal papilla. And this is pretty much what it would look like microscopically, the glandular tissue. Now here we can see a villus. See, we have the circular folds. On each of those folds, we have the villus. And if we look close at one villus, we have the columnar epithelium, and that columnar epithelium has microvilli on its surface. So again, we have surface area upon surface area upon surface area. Now looking inside the villus, we again have this lymphatic vessel, which we call a lacteal, which, as I've said before, will absorb certain fats. And we also have capillaries in there as well. And so other nutrients like amino acids, glucose, some of the other fats will get absorbed into the capillaries, and then, like I said, the other fats will get absorbed into the lacteal. Oh, let me go back here again. Here again are the circular folds. The epithelium again is going to be made up of that simple columnar epithelium. We have our submucosal layer, our circular muscle layer, our longitudinal muscle layer, and the serosa. I kind of mentioned that early on, so just kind of a little review there. Okay, let's talk about the liver. The lobes of the liver, we're going to have major lobes and minor lobes. The major lobe is going to be the left and right lobe of the liver. Now, if we tip the liver up and look under the hood here, we're going to have the quadrate lobe, which is near the gallbladder, and down here, the caudate lobe, which is going to be more caudal. Okay, so the caudate lobe. And then we can see the inferior vena cava kind of pushing past the caudate lobe. And then the ducts, we have the common hepatic duct, and this is not a good picture for it, so we'll go back to another picture in a little bit. Also, I have a separate video that just looks at the ducts. But we have the common hepatic duct coming from the liver. Coming from the gallbladder, we have what's called the cystic duct. Now, a cyst is a fluid-filled sac, right? And so the gallbladder is what? It's a fluid-filled sac, okay? So cysto, again, refers to that, that sac containing a fluid. Think about if you have a bladder infection. We call that a cystitis, because again, a cysto being a fluid-filled sac. So since the gallbladder fits that bill, the duct coming off of it is going to be called the cystic duct. And so the common hepatic duct and the cystic duct come together to form the common bile duct. And then again, it joins at the pancreatic, with the pancreatic duct at the hepatopancreatic ampulla. And so the liver is one big biological factory. It does a lot of stuff. And one of those things is bile production. And what bile is salts which emulsify fats, and they contain pigments such as bilirubin. Gives it kind of that green appearance. Now, the gallbladder simply stores the bile that the liver makes. So if you have your gallbladder taken out, do you still make bile? And the answer is yes, because it's the liver that makes the bile. And when we talk about emulsifying fats, what does that mean? I don't know if you've ever seen these commercials where, you know, they're washing dishes and now all this grease is floating in the water and they still have another glass to wash. So they take the dishwashing liquid and they put a couple of drops into the water and all of a sudden you see all that grease disappear off the top of the water. Well, soap is an emulsifier. What it does is it takes that fat and it breaks it down into little microscopic individual particles called micelles. And that's what the bile does. Okay, it takes the fat that you ingest, it breaks it down into tiny little particles that we're able to absorb then. And so if you have your gallbladder taken out, you're still making bile. However, depending on what you eat, you may not have enough bile available to handle all of that fat. It's like washing dishes. You've got nice soapy water, and that's taking care of a lot of, grease on pans and things like that. But you'll get to a point where it's just not doing the job because there's not enough soap there. You'd have to add more soap. Well, same thing here with your gallbladder out. You don't have the surplus of bile stored up to handle the amount of fat that you might eat if you eat a heavy fat meal. So what happens to that fat? Well, it goes down through your digestive system like it's at one of those Wisconsin water parks, and, well, you might need a change of shorts. I'll just put it that way. Okay. So again, the liver is what actually makes the bile. The liver also stores things. It stores glycogen. Remember, that's a quick source of glucose when your blood sugar is starting to drop. It stores fat. It stores vitamins, especially the fat-soluble vitamins, A, D, E, and K. It stores copper, and it also stores iron. And when you're making red blood cells, this is one of the sources where iron can be gotten from. Now, they used to tell you if you had iron deficiency, anemia, to eat liver. Okay, I don't know if they still recommend that or not, but they used to tell you to eat liver because liver does contain quite a bit of iron. Going back to vitamins, now there's water-soluble vitamins and there are lipid-soluble vitamins. The B vitamins, for the most part, are water-soluble. Okay. Couple of the B vitamins you don't want to take large quantities of, but most of the B vitamins, if you take a large dose of it, it's not going to hurt you. All it will do is make your pee look very fluorescent. Okay, very yellow. So you get rid of it. You don't store up toxic amounts of it. Actually, I think it's B9 that you can start to cause some nerve damage. Don't hold me to that, but I think it's B9. We use it to treat like peripheral neuropathies, but in excess it can cause peripheral neuropathies. But when we go to the lipid-soluble vitamins, you can build up toxic levels of those. You don't want a megadose on those. A is one of those, vitamin A, where you can actually develop a toxic amount of vitamin A. This is why it's best to take beta carotene, for instance, because then the body can take the beta carotene and convert it into how much A it needs. Okay, so the beta carotene is not going to bother you, but that can be converted into A. But large amounts of vitamin A, toxic. So if you decide, this healthcare thing is not for me, I'm going to become an ice road trucker, and you're out there on the tundra, driving around, and maybe you're, kind of lost, and there's a blizzard, and you don't know where you're going, you're starting to get hungry, and you accidentally clip that polar bear, thump, oops. Okay, and you decide, I'm going to have polar bear steaks, but liver's pretty tasty, and I know all of you are going, yuck. I like liver. It's good. With A1 sauce, because if you don't use A1 sauce, then it tastes like liver. But anyway, if you go ahead and eat that polar bear liver, guess what will happen? You will die. You will die of vitamin A toxicity. And I have heard that most carnivores do have large amounts of vitamin A stored up in the liver, so their livers tend to be toxic. So the livers that we eat, like chicken livers and calf liver and all that stuff. Those are from grazers. Okay, so those are from vegetarian animals. So don't eat carnivore livers. And I know you're saying, no problem. I won't be eating any livers. Anyway, so other functions of the liver: nutrient interconversion. Basically, it's going to take nutrients and convert them into usable nutrients and also detoxify them. So the hepatocytes remove ammonia, which is a byproduct of breaking down proteins, and it converts that ammonia, which is very toxic, into urea. Now, urea in high levels is also toxic, but it's less toxic than the ammonia. And then we tend to urinate that out. Okay, so the urea. And now, back in my day, we used cloth diapers with kids, and used to have a big diaper pail. So, baby peed their diaper. You take that diaper off, throw it in the diaper pail, throw on a new cloth diaper, and hope you don't stick them with the big diaper pins, which happens a lot, actually. But I didn't have kids, but I just remember with my brother, and it wasn't that difficult to accidentally stick them with those big diaper pins. So, not fun. Or you'd stick yourself. They'd go through your finger. But anyway, those diaper pails, when the diapers sat for a while in there, before you washed them, they would start to smell like ammonia. And that's because the urea in the urine would start to break back down into ammonia. Okay. And then phagocytosis, we have some Kupffer cells, which are phagocytes. And remember, they started off as monocytes in the bloodstream, but when they go to live in the liver, they become the Kupffer cells, and they phagocytize worn out and dying red and white blood cells, and also some bacteria. And then synthesis. The liver makes albumins, fibrinogen, globulins, heparin, clotting factors. Let's think about somebody with liver disease. If you're not making enough albumin, remember, albumin has a very strong osmotic pull. It helps keep the fluids within your blood vessels. Okay? And if you're not keeping the fluids within the blood vessels, they leak out into the tissues. So in somebody with liver disease, oftentimes you'll see swelling. As a matter of fact, a lot of that fluid can start to accumulate in the abdominal cavity, and we call that ascites. And they have to stick a kind of a large needle into the abdominal cavity and drain off that large amount of fluid. Also, fibrinogen. You're going to see people with liver disease have a lot of bruises, because they're not going to be making the fibrinogen. Also, it mentions clotting factors. So they're not going to be making as many clotting factors as well. And they're also going to be more susceptible to illness because, again, we are reducing the amount of globulins that are being produced, and that's part of the immune system as well. And this just shows the blood and bile flow. And so we have blood coming from the aorta. We have oxygen-rich blood going to the liver via the hepatic artery, and oxygen-rich blood going to the small intestine. Okay. Now, nutrient-rich oxygen-poor blood is going to go into the hepatic portal vein and to the liver for processing. From the liver, it goes from the hepatic veins, dumps into the inferior vena cava, and now we have all that nutrient-rich blood that's going to be then sent to the lungs to pick up oxygen and sent throughout the body to distribute that oxygen and distribute the nutrients. Now from the liver, again, we go from the hepatic ducts into the common hepatic duct, and then finally into the cystic duct, well, from the cystic duct into the common hepatic duct. And so eventually that bile enters the intestines, and it's within the intestines that it gets broken down. And basically we wind up with that brown color that you see in feces. Okay, looking at the duct system again. We have the hepatic ducts coming down into the common hepatic duct. So this is the common hepatic duct. Here's the cystic duct coming off the gallbladder, combines to form the common bile duct, hooking up with the pancreatic duct, forming the hepatopancreatic ampulla, and then the major duodenal papilla. So that's the flow of bile. One thing I want to mention about the pancreas is, again, it makes a lot of digestive enzymes. Some of those are proteases. And I had an instructor who actually just a month or so ago from the time of this taping, I just found out passed away. He was one of my favorite instructors. He was my chest x-ray radiology instructor. And so we also lived in the same town, so I'd run into him. Great guy. But one day he became very sick, and he started bleeding internally. Rushed him to the hospital. What they found out was he developed a little cyst on his pancreas, on the tail of his pancreas. Look how close that is to the spleen. So when those digestive enzymes ruptured, they began digesting the spleen. The spleen is very blood rich. Okay, so it started digesting the spleen, and that is what caused him to start bleeding internally. Luckily, he got in time. They were able to remove the spleen and repair where the cyst had ruptured in the pancreas. So, but that was a close call for him. So going back to the gallbladder, bile is stored and concentrated in the gallbladder. It's stimulated by cholecystokinin, or CCK as we abbreviate it, and also vagal stimulation, in other words, stimulation from the vagus nerve. And when the gallbladder contracts, it's going to dump bile into the small intestine, more specifically the duodenum. Now, what can happen is you can develop gallstones, and there's different reasons why these gallstones can happen. One is drastic dieting with rapid weight loss. Now, let me be clear on this one. This would be more with low-fat diets, okay? And the reason is if you're on an extremely low-fat diet, then the bile sits there. Then the bile starts to solidify and crystallize, and that's when you wind up with the stones. It's not necessarily true if you're on like a keto-based diet, or like an Atkins-like diet, because you're eating high fats in that case, and so that bile is going to be used. Okay. Now, sometimes people start to have gallbladder problems when they're on higher fat diets, and that's because they've already started developing stones, maybe from trying these other diets, and So when you're eating the higher fat diets, and if you have the gallstones, the gallbladder starts contracting down to try to squeeze out some bile. But think about this: if you have a handful of sharp rocks and you start squeezing them all day long, what's going to happen? Your hands are going to start to hurt, right? Same thing here. Now, can we see gallstones on an X-ray? Yes or no? Well, actually, you cannot see gallstones on an X-ray. And when I say you can't see gallstones on an X-ray, there's always exceptions. Occasionally, gallstones can become calcified. That's more rare. Those could be seen on an X-ray. But your typical gallstone is just made of bile, not calcium, and X-rays will pass through it, so you won't see gallstones. So we typically use ultrasound to look for gallstones in the gallbladder. Okay, the pancreas. Pancreas is, you get two glands for your money. It's endocrine, it's exocrine. The endocrine function, again, we have the islets of Langerhans, or pancreatic islets, and that produces insulin and glucagon. Insulin, again, lowers blood sugar. Glucagon raises blood sugar. Remember, the glucagon goes to your liver and tells it to break that glycogen down into glucose. And then the exocrine function, the acini, or grape-like clusters, are going to produce the digestive enzymes, which we'll look at in just a second. The regions are the head, body, and tail, as I showed you in the earlier slide. The pancreatic juice, which is a function of the exocrine portion of that gland, is trypsin, chymotrypsin, carboxypeptidase, pancreatic amylase, pancreatic lipase, and enzymes that reduce DNA and ribonucleic acids. And that would be your nucleases. Trypsin and chymotrypsin break down proteins. Carboxypeptidase is going to break down carbohydrates and proteins. The pancreatic amylase is going to break down starch. And starch, by the way, is a complex carbohydrate, so it has to be broken down into smaller carbohydrates, which again eventually will become individual sugars, glucose basically. And then finally, the enzymes that break down the DNA and the ribonucleic acid, again, are going to break those down into the individual nucleic acids that we can utilize to make our own DNA and RNA. And so we see pancreatic secretion control. First of all, we have from the duodenum secretin and cholecystokinin, or CCK. That enters the circulation. That then stimulates the pancreas to start to produce pancreatic juices. And then we can also have vagal stimulation, again from the vagus nerve, to also stimulate that production of pancreatic juices as well. Okay, the large intestine extends from the ileocecal junction here, the ileocecal valve, to that planet near Neptune, you guessed it, Uranus. And it consists of the cecum, which is this blind pouch, the colon, which we have the ascending colon, the transverse colon, and the descending colon and the sigmoid colon. And then we have the rectum right here, and then finally the anal canal. Now, movement is very sluggish through the colon. We're looking at about 18 to 24 hours. And some other anatomy that we want to look at here is we have this, which is called the tinea coli. Tinea means tape, like Scotch tape, a roll of tape. This kind of looks like tape on the colon. So this is why we call it the tinea coli. Okay, so tinea coli. We also have these little outpouchings that are called haustra. We have these epiploic appendages, which are basically little fat pieces that are hanging down. At this one bend here, this is called the right colic flexure. It's also called the hepatic flexure because right here is where the liver sits. And this would be the left colic flexure or splenic flexure because the spleen is over in this area. Okay. And then when we come down here at the anal canal, we're going to have an internal anal sphincter and an external anal sphincter. The internal anal sphincter is going to be made of smooth muscle. That's important because smooth muscle is involuntary. We have no conscious control over smooth muscle. So as the rectum distends, the internal anal sphincter is going to then relax. Well, that could be a big problem, right? So the external anal sphincter is going to be skeletal muscle. And that's what is going to hold everything back until you're ready to defecate. So, and that's why with children, it takes a while for muscle coordination. And so it takes them a while to learn to have control over the skeletal muscle of that external anal sphincter. Now, if there is any type of neurological damage where you might lose the function of that skeletal muscle, this is what can then lead to incontinence. So, and let me go back. One thing I forgot to mention, hanging off the cecum is the vermiform appendix. Okay, we usually just call it the appendix, but we have several appendices. But in this case, vermiform, vermis means worm, form means shaped. So this is the worm-shaped appendix. So again, the cecum is a blind sac, and the vermiform appendix is attached to it. The colon, as I mentioned, we have the ascending colon, the transverse colon, the descending colon, and the sigmoid colon in that order. The rectum is a straight muscular tube. Rectus means straight. Like, remember the rectus abdominis muscles? Those fibers run straight up and down. In this case, the rectum is named that because it's a straight muscular tube. And then the anal canal, as I mentioned, the internal anal sphincter is smooth muscle. The external anal sphincter is skeletal muscle. And then hemorrhoids are vein enlargements or inflammation. Secretion of the large intestine. Mucus provides protection. We have parasympathetic stimulation, which increases the rate of goblet cell secretion. So we have a lot of these goblet cells found within the colon, and it does make quite a bit of mucus. Some pumps that we find in the large intestine, which will exchange bicarbonate ion for chloride ion, and it'll also exchange sodium ion for hydrogen ion. And then bacterial action produces gases called flatus. And it also has a whole bunch of other words too, which I'm sure you're thinking right now. And so we can see, again, the tinea coli, the hostra, which are the outpouchings, the little epiploic appendages hanging down. And then you can see that the large intestine is a little more flat. While it looks like it has villi, its surface area is not quite as extensive as like the small intestine. Okay. While there are things that get absorbed in the large intestine, most of that digestive absorption is taking place in the small intestine. Okay. But we can see lymphatic nodules here. We see the crypts. We see the goblet cells within the crypts. And then again, our circular and longitudinal muscular layers. So movement in the large intestine. We have mass movement, which is common after meals. This is why, like if you're feeding a baby, right? Food's going in one end and what's coming out the other end. That is not the same what's going in as you're putting it in. It's not just coming right out the other side. That's going to stimulate mass movement to move what's in the digestive tract out to make room for more. Same thing if you have an animal, such as a dog, for instance. You feed the dog. What does the dog have to do as soon as he's done eating? You have to let him out. Again, that's because of these reflexes, which then stimulate this mass movement. So the local reflexes, we're going to have the gastrocolic reflex, which is initiated by the stomach, and then the duodenocolic reflex, which is initiated by the duodenum. And then finally, we have the defecation reflex, which is distension of the rectal walls by the feces, then are going to relax that internal anal sphincter, and then it's up to you when to relax that external anal sphincter. And then we have defecation, usually accompanied by voluntary movements to expel feces through abdominal cavity pressure caused by inspiration. That's that Valsalva maneuver I talked about in the respiratory lecture. So Valsalva maneuver, again, is exhaling against a closed glottis, and that is going to increase intra-abdominal as well as intrathoracic pressures, but that helps to mechanically push feces out during defecation. What a gross topic. And so let's just take a look at a little video for the reflexes in the colon.
The presence of food in the stomach and chyme in the duodenum stimulates mass movements in the colon. Mass movements are integrated by the enteric plexus. They propel the contents of the colon toward the rectum. The presence of feces in the rectum stimulates parasympathetic and local reflexes that result in defecation.
Now looking at digestion, absorption, and transport. Digestion is the breakdown of food molecules for absorption into the circulation. Mechanical digestion breaks large particles of food into smaller ones, and that would be such as chewing. Again, we're breaking it down into smaller particles to increase its surface area so that mechanical digestion can break the covalent bonds with enzymes. So the more enzymes that can, attack that particle of food, the more complete breakdown you'll have. So chew your food well. Absorption and transport, molecules are moved out of the digestive tract and into circulation for distribution throughout the body. So looking at carbohydrates, which consist of starches, glycogen, sucrose, lactose, glucose, and fructose. And actually, let's look at those, sucrose, lactose, glucose, fructose. First of all, sucrose. Sucrose it's a disaccharide, which means we can't absorb that as is. We're going to have to break it down. Sucrose is your typical table sugar. And so what is sucrose made out of? Well, it's made of glucose and fructose. So once we break down the glucose and the fructose, take the sucrose and break it down into glucose and fructose, then we can absorb that glucose or that fructose for energy. The next disaccharide is going to be lactose. Now, you've probably heard of lactose. If you're lactose intolerant, you know what to avoid, right? Dairy products. We find lactose in milk. So what's lactose made out of? It's made from glucose and galactose. Now, how do I remember that? I think of, okay, lactose is found in milk. Where do we live in this universe? We live in the Milky Way galaxy. So that's how I remember galaxy, galactose. Okay, well, that's the way I remember it anyway. So glucose and galactose gives us lactose. And then there's maltose. And maltose is made from glucose and glucose. As a matter of fact, there's a cereal, which I didn't think they made anymore, but I think they still do. Malto meal, which of course is going to be made of maltose. That's almost like giving pure sugar because you break maltose apart. Again, you've got glucose and glucose. So holy cow. Anyway, so once we get it broken down into a monosaccharide, then we can transport it into the cell and then back out of the cell into the capillary. So glucose is transported to cells requiring energy, and insulin influences the rate of transport. How it influences the rate of transport is that we have these GLUT receptors, these glucose transport receptors, that the insulin helps to install into the cell. The more of these receptors that are plugged into the cell, the more glucose it's going to transport. And that transport is going to be facilitated diffusion. So it doesn't take energy, it's diffusion, but it's facilitated diffusion. Okay, so we see here that to get it inside the cell to begin with, we're going to use a secondary active transport to bring it into the cell. Okay, so that will require some energy because it's active transport. But once we get it in the cell, it's facilitated diffusion, which is passive, to bring it out of the cell and then through regular diffusion into the capillary. And it goes off to the liver. And then our lipids, which include triglycerides, phospholipids, steroids, and fat-soluble vitamins. Emulsification breaks down the large particle droplets in the smaller ones. And remember I said bile is the emulsifier. Just to give you another idea of where you're going to find emulsifiers. Next time you have a candy bar, you know, like a Snickers or a Milky Way, one of those chocolate ones, take a look at the back. You'll see something called lecithin. And lecithin is an emulsifier, and that's what keeps the fats, the chocolate fats and what have you, nicely spread out and even within the chocolate. When the lecithin starts to break down, that's when you start to notice the different coloration of the chocolate. Like if it's an older chocolate bar, you'll see stuff, mainly fats, starting to bloom on the outside of the chocolate. I don't think there's anything wrong with that, but again, it just keeps it mixed in there. So again, it's an emulsifier. So we can see here, and these are the micelles I was talking about earlier. This would be an example of a micelle. This is also, remember I talked about soap, how soap works? Soap forms these little micelles around either fat or around dirt. Okay, so that's how an actual soap will work. And it forms, remember the cells are phospholipid bilayers? These little micelles are actually phospholipid monolayers. Okay, so your cells are like a soap bubble. So here's the little soap. In this case, the bile is the soap. And if anybody has a dog and your dog is ever thrown up on the carpet, and there's some green there, okay, they threw up some bile, and if you try to clean it, you're going to notice it's slippery and soapy, okay, because it's pretty much a soap, okay, and that's what breaks down the little fatty acids, okay. So from here, it's going to contact the epithelial plasma membrane, and because your cell membrane is a phospholipid bilayer, it can kind of fuse to it and then allow, by simple diffusion, this fatty acid to pass through the cell membrane. Okay. Hopefully I didn't say cell wall earlier. We're not plants, but through the cell membrane. And it's going to release the triglyceride. So here's the triglyceride. Then the triglyceride gets a protein coat, at which point we call this a chylomicron. And through exocytosis, that chylomicron is spit out and absorbed into the lacteal. And it's then transported through the lymphatic system. And then we have lipoproteins. The types, we have chylomicrons, which enter the lymphatic system through the lacteal. And you can see here that it's made up of 5% cholesterol, 90% triglyceride. Then we have a phospholipid membrane around it, or layer, and then 1% protein. And then we have our VLDLs, which is the very low density lipoproteins. And you can see 60% is triglyceride, 18% is the membrane, 14% cholesterol, 8% protein. Then the two that we're probably more familiar with, the LDL and the HDL. LDL is low density lipoprotein, and that transports cholesterol to cells. And so here's our LDL. And you can see that it has a phospholipid that makes up about 20%. Triglycerides make up 10%, cholesterol 45%, and proteins about 25%. So this transports cholesterol to the cells. And why did the cells need cholesterol? Well, it needs it to make the cell membrane. Remember, the cell membrane is made up of not only phospholipids, but also cholesterol to give it the integrity. Otherwise, you've got a soap bubble, basically. So that cholesterol gives it some structure, some strength, and what have you. But our LDL, is that our good cholesterol or the bad cholesterol? Well, that is the bad cholesterol. Think of L for lousy. Okay, lousy cholesterol. And the truth is, though, none of these are bad. They're all necessary. We need cholesterol. We need cholesterol for cell membranes. We need it to make steroids, for hormones. So it is necessary. It's just that in super large amounts, this can be damaging. And then we have our HDL, which is high density lipoprotein, and this transports cholesterol from the cells to the liver. And so if you have high LDL, but you also have very high HDL, then that's a good thing, because the higher your HDL, the better. I mean, of course, there's limits, but within the normal range, a higher HDL is good, because that's transporting the excess cholesterol from the cells to the liver. Okay. And so HDL, we usually say that's your good cholesterol. And that can be increased through diet, through exercise, or what have you, can raise your HDLs. And then proteins. Pepsins break down proteins into smaller polypeptide chains. And basically, we have to get it down to the amino acids. So these proteolytic enzymes produce the small peptide chains, the dipeptides, the tripeptides, and again, down to amino acids. The amino acids are brought into the cell through secondary active transport. And through active transport, it's brought out of the cell and it goes into the capillaries to the liver for processing. Water and ions. Water can move either direction across the wall of the small intestine, depending on osmotic gradients. And ions, such as sodium, potassium, calcium, magnesium, phosphate, are actively transported. So let's take a look at water. We ingest about 2 liters of water, as liquids, per day. Then salivary gland secretion, we secrete about 1 liter of saliva a day. Wow, think about that. 1 liter of saliva. That would be half of those 2-liter bottles of spit. That's pretty gross. Anyway, gastric secretions, about 2 liters. So again, a 2-liter bottle worth of gastric secretion. Eesh. Pancreatic secretions, about 1.2 liters. Bile is about 0.7 liters. The small intestine secretes about 2 liters. Okay, so that's a lot of liquid in the digestive tract. But where does it go? Okay, 92% is absorbed in the small intestine. 6 to 7% is absorbed in the large intestine. And so basically we kind of have a balance between ingestion or secretion and absorption. Okay, and so 1% is what is lost through feces. So ingested plus secreted minus absorbed is going to give us that 1%. Okay. The effects of aging. We get a decrease in the mucus layer. Again, we can see that can lead to ulcers and such. Also colitis. We have a decrease in connective tissue. Also a decrease in muscle and secretions. We have an increase susceptible to infections and toxins, increase in ulcerations and cancers. As far as the colon goes, the integrity of the colon wall starts to diminish and you develop little pockets or outpouchings, which are called diverticuli. And so you'll have the condition of diverticulosis. And then if those become inflamed, then we have diverticulitis. And so there's a lot of things that go on with this. Some of these things can be screened for and prevented. So if you have a family history of colon cancer, make sure you're getting a colonoscopy and get that checked. Because caught early, then there's something they can do about it.